So first we'll hear from Alan Wolfe. So Alan's professor of microbiology and immunology and co-director of the Loyola Urinary Education and Research Collaborative, where he will talk about an update on the urobiome. And then afterwards, we'll hear from Maria Hadjifrangiskou, who's from Vanderbilt. And we'll hopefully have some time to really kind of hear what they say and have some discussion about what I'm sure are going to be really awesome topics.
Right. So my title's terribly uncreative and a little bit broad. So let me tell you what I'm specifically going to talk about. First, I'm going to quickly go over a couple recently published papers and give you the high points on them. Then I'm going to talk about our attempt to figure out how to do antibiotic susceptibility testing on fastidious and anaerobic urobiome species. And then a clinical study that we're finishing, that we're getting ready to submit, asking what happens to the urogenital microbiome following urogynecological surgery, and if time permits, some of our work on the mouse urogenital microbiome. So here's the first two papers. They're in Frontiers in Urology. You can access them, but they're not yet listed in PubMed. I think it's because the issue is not completed. So the first one is called Tarnished Gold, the Standard Urine Culture, Reassessing the Characteristics of a Criterion Standard for Detecting Urinary Microbes. And this is co-authored by Linda Brubaker, Toby Chai, Harry Horsley, and Raj Kashria, and Bob Moreland. Here are the major points. First, we noted that diagnosis and treatment of UTIs has remained stagnant, that diagnostic tests haven't advanced for decades, that development of novel and accurate UTI tests have not been a priority, in part due to the general perception that UTI care is already sufficient. Then we note the importance of improving UTI diagnostic testing so that we can improve treatment outcomes. We specifically outline the limitations of the standard urine culture, or what I call SUC, because it does, that's used by clinical microbiology laboratories, explaining clearly why these methods cannot be considered the gold standard, primarily because they underreport most of the urinary microbes, even some of the acknowledged and many of the emerging uropathogens. Importantly, we don't recommend the abandonment of this test because there's no universally accepted alternative, but we strongly encourage the development of new and improved tests that can both improve outcomes and preserve antibiotic stewardship. I encourage you to read it. My co-authors were fantastic. Their thoughts were really quite insightful. The second one is Beyond the Usual Suspects, Emerging Uropathogens in the Microbiome Age. And with the exception of Catherine Putanti and Linda Brubaker, everybody else who's an author is a member of my group. Many of them are junior, and this was an opportunity for them to write up part of this paper. They picked their favorite emerging uropathogen and dug into the literature. And then Bob Moreland and Brian Choi and I sort of try to tie it all together. And so this is, again, the major points are, we set out to highlight a set of poorly understood emerging and suspected uropathogens, to generate momentum for studies to investigate the pathophysiology of these microbes, to identify risk factors and improve antibiotic surveillance. And it's especially true for those species that don't have CLSI standards. So that's the agency that sets the standards for antibiotic susceptibility testing. So we focused on the lesser known microbes, members of the families Aerococcaceae, Actinomycetaceae, Bifidobacteriaceae, members of the strep anginosus group, anaerobic orders Eubacterialis and Bacteroidealis, and Enterococcus faecalis, because urinary isolates differ from those of other human niches. We have a paper that was recently submitted. It's a phylogenomics paper showing that E. faecalis, the genomes of E. faecalis isolates from the urinary tract, in some respects are distinct from the genomes of isolates from other niches. All of these are either underreported or not reported by SUC, but they're routinely detected by sequencing and metaculture approaches like our EQUIC. And again, I recommend that you read it. Like I said, it highlights a bunch of bugs that many of you are only now becoming familiar with. Some of them you can actually pronounce. And so now I want to talk about trying to set up testing for antibiotic susceptibility for species that simply don't grow under the standard. So here's a little bit of the background, right? So the ecological niches of the urinary tract range from aerobic to anaerobic. But CLSI in the States and UCAST in Europe, their standards only evaluate aerobic conditions of mostly non-fastidious urinary microbes. The standard medium for non-fastidious microbes is cation-adjusted Miller-Hittenbroth, or MHB. Recently, within the last year or two, CLSI put up a recommendation that you might work with fastidious organisms by adding 5% lysed horse blood, or LHB, to the MHB. The problem is that blood renders the media opaque, and so you can't take ODs. So on the right, you can see that MHB plus LHB is pretty opaque. On the left is MHB, and our solution is the one in the middle. It's NYC3. We didn't invent it. We learned about it because we were trying to grow Gardnerella, and I think it was Mandy Lewis who told us, oh, you to try NYC3. But it's transparent, as you can see, because it replaces the lysed horse blood with horse serum. We don't know what's in horse serum, but it seems to be magic. It's calf serum doesn't work. There's something special about horse serum that probably is more like human serum. So anyway, the first thing we wanted to do is to see whether or not NYC3 is permissible for the growth of most of our species. And that's the red bar, and you can see that it grows the usual suspects: E. coli, Pseudomonas, Enterococcus, Staph aureus, and so on. But it also grows Streptococcus pneumoniae, which will not grow in MHB. That's the gray bar. You can see it's missing here. Or the lactobacilli, Lactobacillus crispatus, for example, will not grow on MHB. Its preferred medium is MRS, but it will grow in NYC3, and so will virtually all the rest. Now, we're not talking about, and by the way, the blue bar, of course, is the MHB with LHB. So we're not arguing that you use NYC3 for everything. I mean, if something grows pretty well in MHB, the standard, you should use it. Anyway, this was the design of our study. The first, we took the standard species, CLSI standard species in their standard medium. So these are ATCC strains. And we obtained the MICs and found that they were in the CLSI range. So that's important, proves that we can do the assay properly. Then we took the standard species and grew them in NYC3 and got our antibiotic susceptibility profiles and compared those results to those in the standard medium. Once we had validated our NYC3 approach, we could then do high throughput 96 well dilution series assays on fastidious and anaerobic species in the NYC3 medium. So here's the evidence that validates NYC3 as a growth medium for antibiotic susceptibility testing. Here are the 5 standard usual suspects on the left side, and then a set of antibiotics across the top. And if the cell is green, it means that both the values in NYC3 growth medium and in MHB growth medium were within the CLSI QC range. So there are 3 scenarios where we're outside the range, where NYC3 was outside the range. It's not perfect, it's not a one-size-fits-all, but it does work. Our NYC3 approach works pretty much as well as MHB. So NYC3 does the job. So then we applied this to a subset of urinary bugs that are fastidious. And we did this on a number of different species, a number of different isolates of each of those species, with a number of different antibiotics that are typically used in the clinic. And what I'm going to do now, and we did this for 48 hours. We grew them for 48 hours and 5% CO2 at 37 degrees with shaking. And I'm going to highlight the vancomycin set just so that I can blow it up. So we did Actinomyces tercensis, which I think now is called Chalier tercensis, Actinotignum, Timonens, Aerococcus urinea, Carini, and a couple of staphs and streps, multiple different numbers of isolates. And what you can see is that we can, in fact, get MICs. We did the same thing with anaerobic species and Anaerococcus, Gardnerellus, the lactobacilli. Now, I know that they're typically, beneficial bacteria, but we wanted to see about not just, the suspected pathogens, but pretty much everything that can grow, that we can find in the bladder. Peptoniphelus and Propionibacterium. And again, there's a fewer number of isolates that we've tested, but we can, in fact, get MICs. And this was for 3 days under anaerobic conditions. We have an anaerobic chamber at 37 degrees and shaking. So the conclusions are that NYC3 is a valid testing medium for fastidious and anaerobic microbes. NYC3 is translatable to the standard MHB approach. It's within the CLSI QC ranges, and it can be used in a high throughput 96 well format and spectroscopy. And the paper is almost ready for submission. We're going to submit it to the Journal of Clinical Microbiology. All right, now I want to talk to you about a clinical study that we've been doing with Beth Miller's team in the urogynecology section of Loyola. We wanted to know what happens to the urogenital microbiota during and after urogynecological surgery. There's a lot that happens to a patient when they're undergoing surgery, and we were pretty sure that the microbiota would be disrupted. We wanted to know how resilient it was. And so, yeah, that's the question: is the urogenital microbiota resilient after surgery? And the sub questions were: does the UG microbiota return to preoperative baseline after surgery? If so, how rapidly does it return to baseline? And does the recovery process differ in pre and postmenopausal females? So this was done, all the recruitment was done between in 21, 22, and the very beginning of 23. Some of the inclusion criteria was that they had a vaginal preparation with chlorhexidine. They were, because they're going into surgery, they had systemic antibiotics. They had to have a placement of a Foley catheter or cystoscopy during the case. Few other, there are certainly some exclusion criteria, but in the interest of time, you can read it when the paper is accepted, which I hope it will be. So we took catheterized urine specimens and vaginal swabs. So the catheterized urines are bladder, the vaginal swabs are genital, and we did this longitudinally. We took samples 4 times. One was before surgery, on the day of surgery, then immediately after surgery, okay, then at the 3-week post-op visit, and then 12-week post-operative visit. The samples were kept cold until they got to my lab. We preserved them as assay assure and threw them in the minus 80 degree incubator. We did the typical DNA extractions that we've been doing in my lab. We used the V4, we amplified the V4 region. We sequenced it with Illumina, short read Illumina technology on a NextSeq 2000. We trimmed the reads. We used DADA2 to determine the amplicon sequence variants, and then we used BLCA, Chung-Fung Dung's algorithm for determining taxonomy. So I'm just going to give you very, very processed data in the interest of time. This is a linear mixed model of alpha diversity measures. We have the 4 time points across the top. We have postmenopausal and premenopausal. This is the bladder microbiome. We're using Shannon here, which is a composite measure of alpha diversity, and then the p-values are over. You can see that the p-values for menopausal status and for time are both significant. So what you can see down here is the plotting of these, the numbers that are in the pink and blue boxes, and what you can see is that indeed there's a dramatic increase in alpha diversity from pre to immediately post-op, and then they drop down by 3 weeks, and they're pretty stable at 12 weeks. Okay, so there's clearly a disruption, and there's clearly in terms of a alpha diversity that there's some sort of recovery. Now, we wanted to ask the question about whether, let's go back. The alpha diversity doesn't tell you what the composition is. It just tells you richness, abundance, and evenness. So how many bugs there are, how many different taxa there are, what's their relative abundance, and what's their distribution. We wanted to ask, so you can start with a particular alpha diversity and end with the same alpha diversity. The composition could be completely different. So we used Jensen-Shannon divergence algorithm to determine whether or not the community went back to baseline by 12 weeks or had gone to some different composition. So each subsequent time point, all the subsequent time points are compared to the preoperative time point. So the communities that we saw in the other 3 time points are compared to the preoperative community. For each individual, you're scored from 0 to 1. The larger values are in red, they're dissimilar, and the green ones are more similar. So on the left, we have the premenopausal women, postmenopausal females are on the right. We had a number of different participants in both groups. And really, I just want you to see is that whereas immediately post, for the premenopausal females, they generally come back to, well, in most cases, sort of a brownish color or a green color, whereas the postmenopausal women, they are red. So what happens is how we interpret this is the premenopausal female microbiota return to a composition that resembles the baseline in most cases. And that in most cases, the postmenopausal composition differed following surgery than at baseline or before surgery. So here's another way to look at that. This is set up similarly. You can clearly see that immediately post-operative, 3 weeks and 12 weeks for the premenopausal women, there's really no change in the JSD values, but the values for the postmenopausal women increase and they stay high. So the conclusions here is that the alpha diversity measures return to baseline at 3 weeks and remain in equilibrium at 12 weeks. For the urogenital microbiota, we did this for the vaginal microbiome as well, that the composition, stability, and process of recovery differ between pre- and postmenopausal females. Now, finally, and this is a paper that's about to be submitted as well. Finally, I want to show you a little bit of work that we've been doing on trying to identify the mouse, characterize the mouse urogenital microbiome. Now, there's a couple of people that I know are listening in who know that I've been trying to do this for a while. we've actually made a little progress recently. This is work that I've been doing with Katie Foster at Pittsburgh. The typical mouse model of UTI requires that you use the right mouse background, and you give them a bolus of, I don't know, 10 to the 7th colony-forming units of E. coli or Proteus or whatever your poison is. And I think the reason that one has to use a bolus is because of colonization resistance. If the human has a urobiome, then I'm sure the mouse must as well. And so we wanted to characterize it so we can figure out how we might be able to improve mouse models, at least to be able to interpret the data, mouse model data, in the context of the fact that there is indeed a mouse urobiome. So first, simple questions. Is there a mouse urobiome? If so, what taxa are predominant? And how sensitive is it to environmental conditions? So we got urine expressed from 2 sets of mice, obtained from the same source, housed in 2 different facilities in 2 different cities at 2 different times, and we sequenced different V regions, but they were all analyzed simultaneously with the same pipeline. Okay, so here at the genus level, here is heat map. One of the sets, these mice were milked for urine this summer, and we sequenced the V4 region. This is Katie Foster as an attending at Pittsburgh, and MedStar was Katie Foster as a fellow at MedStar, and it was sequenced several years back, and the facility that did the sequencing sequenced the V3 to V4 region. Again, as I said, we then took all of those data, put them together, analyze them at the same time. And I don't expect you to be reading the genus names, but let me tell you that the most prevalent and abundant genera, the ones that are up in the top left, are Staphylococcus, Mammillacoccus, Enterococcus, and Ligulactobacillus. And from Pittsburgh, those are ranked 1, 2, 3, and 7. And at MedStar, they're ranked 1, 6, 3, and 2. So we're very satisfied that, first of all, we could indeed get data out of mouse urine that we saw, to a large extent, very similar communities, and that the environment didn't seem to matter that much. Now, this is different from the mouse gut, where if they're in 2 different rooms or different cages, the mouse gut might actually be different, even if you're using the same types of mice. So that bodes well if this holds up for urobiome work in the mouse, because maybe it's not nearly the mouse bladder. The urogenital mouse microbiome may be considerably less sensitive to environmental factors, because I don't think they eat their fellows' urine. They do eat feces. We tried to get to the species level. We didn't have enough information using V4, but with the V3 to V4 region, we did get a little bit of data. And we identified sort of the top is Ephicalis, Staphylococcus xylosis, and Lactobacillus johnsonii. And interestingly, and this is for you, John Barish, we actually cultured these bacteria in previous attempts to study the mouse urobiome. John provided us with some urine several years back. We did EQUAC on them, but we never had enough. We never had enough mice, and we didn't have enough depth to be able to say very much using that approach. But it's promising that 3 very different approaches, 2 sequencing and 1 culture, provided us with relatively similar results. So I'm a little optimistic, and we're getting ready to write this up. Before I'm finished now, I just want to highlight the fact that In the past, we've had at the CMI International Microbiome meeting, there's a Urobiome Day, the third day. In 2024, we're going to do it again, except that for reasons that are sort of outside our control, we're now integrated with the other 2 days. I would like to suggest that this is a really good place to do 2 things. One, to find out about general microbiome work going on in the world. Rob Knight is one of the organizers. He brings in the very best from all over the world. I've learned an awful lot from sitting through the first 2 days, and to have an opportunity to listen to some of the leaders in the Urobiome community give talks. And we're working on having breakout sessions. I can't say that they're going to happen, but I suspect they will. At the very least, we all can get together and network, talk about each other's works. So I mostly sit behind this desk. I'm co-director of the Loyola Urinary Education and Research Collaborative, along with Beth Miller. All the people that are in bold are current members. The ones that are not in bold are recent people that have recently left us, but were involved in some of the work that I just described. Our funding at the bottom right, and I'm happy to take any questions.
Awesome. That was a great talk, Alan. We're going to hold questions until the end, and we can have like a single discussion, if that's all right. And we can move on to our next talk, which will be given by Dr. Maria Hadjifrangiskou, who's Associate Professor in the Division of Molecular Pathogenesis in the Department of Pathology, Microbiology, and Immunology at Vanderbilt University Medical Center. So I'll give the floor over to her.
Thank you all for inviting me to this forum. I enjoy so much being on the CAIRIBU online discussions. And I've titled my talk, "People are convinced now," for the most part, right, Alan, that there is a urobiome, but, what do we do with this? So I decided to start my talk like this because for those of you in the audience who know our work, our lab has traditionally focused on understanding mostly how primary uropathogens like Escherichia coli interact with the host environment and with one another. And we've specifically been really focused on understanding how these uropathogens build these multicellular communities that are known as biofilms and that you see here. But of course, as all of you know from seminal work from Alan's group and others, there is a urobiome, right? So we have started recently to ponder, how does the urobiome interfere with the uropathogen's physiology? More importantly, the bladder physiology, and then how does that change the interaction of the bug with the host in the context of a known urobiome, right? So that's how my lab started to get into the field slowly and shyly, because of this thirst that I have to understand community interactions with my favorite uropathogens. But as you all know, to achieve this, and again, I'm glad we're having this community build, because we need to have a good grasp of the constitution of the urobiome, right? And then how it varies across time, right? What happens across the lifespan, where I feel we need to have some more studies to go in. And this is more for the trainees, right? And Alan alluded to this earlier, but we use the same standard urine culture protocol. Yes, it kind of sucks. You're right, Alan, but it was developed by Edward Haas in the 1950s, and the main goal was to capture the main uropathogens that we all know. But over the next 60 years, right, we've seen the blooming and the completion of the Human Genome Project, the blooming of the microbiome, and then primarily spearheaded by Alan, the discovery of the urobiome, and of course, different methods for extended quantitative urinary culture. Right, so having these tools now, we want to move forward and really start filling the gaps, right? And from looking at the literature throughout the years, there's a few things that we still need to do better on, right? And something that I've been talking with Chris, it would be fantastic, for example, if we start building on the CAIRIBU U-RIG website this lifespan information with regards to what is the ecology of the healthy urobiome. I'm using words like from my friend Megan Beringer here, who's on the audience, right? We need to think about these as ecological niches, and how does the composition of the residents change as the person matures, and how does it change with sex or with a special condition like spina bifida, right? So while a lot has been done and highlighted by Alan earlier, there are still gaps that we need to fill. And I think as a community, we can stand to do this very well. So that was my PSA. So here is a group of circles that I've seen presented in this recent a Urobiome review by Coilela et al. And what here you see is some genera that have been seen across studies in all ages in the Urobiome. And then on the green side, you see ones that are common in young and older individuals. And then the blue and the yellow feature genuses, genera that are unique to the old population or unique to the healthy young population. But again, young here is 20 to 49. So my group identified a gap in the field, which is infants and very, very young individuals. And this, of course, is also fueled by the fact that we have a really great collaboration with pediatric urology here at Vanderbilt. And every year we host a pediatric urology research fellow who has really helped us hone into these gaps that we then proceeded to fill. So what I'm going to highlight here in the next few slides is a paper that is just gotten accepted at MPJ Biofilms and Microbiomes, but we also have it on medRxiv so that you can easily access it. And we also have all the code that we used publicly available on the corresponding students' GitHub websites. So for this study, what we did, we enlisted a cohort of 50 healthy infant males that were undergoing circumcision in the operating room after they were born. So these are a little bit older infants for some reason did not get circumcised at birth. So we're looking at a median age of like 4 to 6 months. So what we did, we collected catheterized samples in the OR, and we aliquoted specimens for extended culture, as well as 16S rRNA amplicon sequencing. We're very, very big on controls, and we also have these publicly available. And what we did, we also collected samples from the saline used to prep the patient, the catheter lubricant, as well as the saline flush that was used to flush the catheter that we used to catheterize the patient. So these also underwent extended culture, and they also underwent 16S rRNA amplicon analysis. And the culture, thankfully, to the naked eye, did not grow anything. But of course, as you can imagine, 16S signatures were obtained. But to our happiness, we saw that these sampling controls were all far away from our patient sample population. So having seen these wonderful controls separating out, then we went in and analyzed what we see in this super young male population that has not been yet characterized. So what I'm showing here is a bird's eye view of the 6 most prevalent genera in the infant male population. And I hope, Alan, I think is already looking at this. You see some interesting common ones, like we see Prevotella. This has been seen in all of the ages that we have seen before. So that's one big one. Lactobacillus as well. But then we see prevalence of staph, Nocardiopsis, and Escherichia. And then in all of them, there's a smattering of other genera that we were able to capture a variety of using extended culture. And again, thank you, Alan, for spearheading the field there. And what I want to draw your attention is this species here that we consistently identified with our extended culture. And on the very top is this Actinodignum species, and specifically Actinotignum shalei. And this drew our attention because we isolated it from like 9 of our patients that had a positive urine culture with extended methods. And again, I want to point your attention this Actinobaculum, it's actually the same genus. It was renamed to Actinotignum later. So if you have attended this CAIRIBU U-RIG before, you know that many people that are in the audience today, like Tanya, study lactobacillus. There's other studies that are focused on strep, like Alan has shown. So we got extremely interested in this actinotignum guy here, because again, it's a very, it's a usual suspect. We've seen, it was like, I've seen you before. So we decided to look at actinotignum specifically. So again, this is from Alan's early paper, and here's actinotignum right there. That's actinobaculum. And this is a quote from Alan's paper in both samples, both suprapubic and catheterized samples. We had Irococcus and actinobaculum, which is actinotignum genus. So if we look at what we know about actinotignum species, we know that the first case was discovered in 2010 for the US. And then the first genome shortly thereafter was published in 2014. In the clinical lab, you use MALDI technologies to identify fingerprints of uropathogens, pathogens in general. And there was one developed for actinotignum schalia in 2016. So the clinical lab has the ability to identify this pathogen now, or potential Urobiome member. So then our question, again, as basic microbiologists wearing back our real hat, our real identity, is what is the role of a Urobiome member like actinotignum schalia in shaping uropathogen-urobiome interactions, UTI progression, and of course, down the road, how does it influence host biology, right? If it's a part of the urobiome healthy bladder. So not much is known about A. schalia. If we run your typical PubMed search, either using actinobaculum or actinotignum, I hope you can appreciate the dearth of results here, 91 and 46. And then if we look at these more specifically, they are a good majority of them are case reports. So we have 55 case reports essentially reporting actinotignum as a potential pathogen. And this is work, a literature search performed by my student Jamisha Francis in the lab, and all the data I'm going to show now is from her hard work. And I hope you can appreciate here, there are a good number of them comes from urinary tract infection, 121 cases. Cystitis, prostatitis is the biggest chunk of those. So it affects, it's an emerging rare uropathogen in older males. Now that kind of triggered us, right? Okay, we see it in the male urobiome early on in life, and then we see it emerge as a uropathogen later. Is there a connection there, right? So a few of the questions we want to ask next is, how prevalent is this across the population? Can we see it interacting with uropathogens like E. coli? And, you know, we shorthand it as UPEC here, but there are other uropathogens we'd like to address as well. And then, of course, first and foremost, can we grow this bug and genetically manipulate it? Because again, there's not much out there. So what Jamisha has set out to do, and we are still collecting data on this, is to figure out how prevalent is this across the population. So our study, which is about to be published, is regional, right? We used VUMC patients and we found this, and we validated the prevalence of actinotignum in microbiome studies from the work of the community, but how prevalent is this across the population? So what we're doing now in collaboration with the VUMC clinics is collecting urine samples from people who don't have a UTI, plate them on CNA and blood agar that are permissive for actinotignum, and looking for morphology that matches to it, as well as collecting the urine for subsequent 16S analysis, because we want to know, if we find it, what does it usually come with the caveat, of course, that these are clean catches and we'll be seeing periurethral flora as well. But this is one of the ways that we figured we can evaluate how much of the population carries this bug. And like I said, our goal is to look at prevalence of this bug on these beginning studies. So please, if you're interested in collaborating with us and you have a good rapport with your clinical microlab, give us a call, because we'd love to extend this geographically beyond the state of Tennessee. But now, to move to the meat of the data here, can we grow actinotignum? And compressing 6 months of work by the student in this single successful slide. Here you see two very common laboratory media, lysogeny broth and brain heart infusion, that all of us use to grow non-fastidious pathogens. And I hope you can, or bacteria, not just pathogens, I'm sorry. And what I hope you can appreciate here is that actinotignum does not like this at all. But then Jamisha, from reading the literature, considered different additives, and one that was really, really successful was a mixture of 1% lipids. So incorporation of lipids in this BHI broth skyrocketed the growth of actinotignum, and we also saw this wonderful increase in colony-forming units. You can see how turbidity can be here misleading, right? We inoculate the broth, but when there's no lipids, the bacteria really don't replicate. They either get lost or like lyse over time or stay stable. But when we feed them lipids, they actually have nice replication. And again, I hope you can appreciate this time scale is much more slow to grow compared to your garden variety non-fastidious E. coli, for example. Jamisha is a scanning electron microscopy aficionado. So what you see here are the first ever images of scanning electron microscopy of actinotignum. She went in and she's able to grow it on a slide. She can actually take measurements. All these little yellow points are bacteria that she's actually painstakingly measured the area, length, width, and circularity of. And we announced that this is truly a coccobacillus, and this is what it looks like. And we're excited to see it also form these microcolonies adorned with what we think is an extracellular matrix. So again, these are the first SEM images of Escherichia, and I'm very proud of Jamisha, who actually spent so much time to do this. So Jamisha then went one step further and asked if lipids allow actinotignum to grow, what specific component of those lipids makes it work? So she went and broke down the 1% lipid mixture from Fisher, and she found palmitic acid, myristic acid, arachidonic acid were parts of the constituents, along with others. She grew the bug in specific concentrations of each, and one of the things that we saw to our amazement was myristic acid and arachidonic acid were sufficient to increase the growth of this bacterium in vitro conditions. So one of the things, and again, here's a picture of the plates at 24 hours. It usually takes 72 hours to see colonies, and we see that with myristic or arachidonic acid, this bug has really nice colonies by 24 hours of incubation on CNA and blood agar. So one of the things in my animation here, when crazy, so I'm going to start with this point. Why are we so excited? A, we can grow it now. We can do some pretty fun stuff to understand its physiology. But number two, it grows on host fatty acids. Well, arachidonic acid specifically caught our attention because from reading a little bit in the literature and attending meetings like the Society for Basic Urologic Research, where I always think, I'm not going to learn anything. But usually, you know, I leave with some nugget of knowledge. And one of these nuggets is that people with BPH, and this was validated in rat models, arachidonic acid is increased by 10-fold. So I'm sitting there thinking, could increase in arachidonic acid coincide with a bloom of actinotignum in older men? So that would be very interesting. That could potentially, you know, be a reason why we see this increase in emergence of this as a uropathogen. On the other hand, another thought is that arachidonic acid is released and typically associated with controlling pathogens, because if pathogens incorporate arachidonic acid or host fatty acids in their membranes, the membranes become more fluid and they become more susceptible to killing. Well, if actinotignum doesn't undergo that because it has evolved to assimilate host fatty acids to its benefit as opposed to its demise, that is one more nugget of information that can lead us to understand how these urobiome members can coexist with their host. So these are all conjectures, of course, based on these basic lab experiments we've done, but these open avenues that we would like to investigate moving further. We also sequenced these actinotignum genomes that we obtained from our cohort. We obtained the core genome and matched it to a reference strain that has been published in the literature and we have in the lab. And again, if I point your attention here to these peacock green colors, there's lipid metabolism pathways that we're now digging hard to evaluate their contribution into the phenotypes that we're seeing. So we are having membrane lipidomics ongoing, so please stay tuned. We'll have data soon, and we're also doing some more fancy cryo-SEM to visualize membrane and potentially capsule, because Jamisha has found some genes in the actinotignum genome that have some identity to capsule genes in other species with similar metabolic requirements like mycobacteria. So this is where we are, and that I hope now you understand why I called the title Now What? You know, that's what we like to play with, the downstream results of urobiomic, community compositions. I hope I have convinced you we have defined one portion of the healthy male urobiome, an isolated representative species. We've developed growth protocols for actinotignum, and we're very happy to share, and we've begun unraveling its physiology. And we've demonstrated reciprocal effects between Escherichia and one uropathogen, UPEG. So I welcome collaborations, of course, in the future. Whatever you guys want to talk about, I'm here. And any input on this super new unpublished work would be greatly appreciated. Thank you.